Welcome to the Genetics series. This video focuses on non-Mendelian inheritance. At the end of this video, students should be able to understand and interpret the variations of non-Mendelian inheritance. Chromosomes hold our DNA as genes that are expressed through proteins, which are inherited from parents by offspring. Each offspring receives one allele from mom and one allele from dad, which were randomly aligned and assorted during meiotic division, resulting in the sperm and egg gametes. Mendelian inheritance postulates that alleles are expressed in either a dominant form or a recessive form. The recessive form of a trait can only be homozygous, where both recessive alleles are present, expressing the recessive phenotype. The dominant form can be found in the homozygous form with both dominant alleles present, as well as in the heterozygous genotype with one dominant and one recessive allele present. Both express the dominant phenotype. These phenotypes follow a predictable pattern. In monohybrid crosses of a 3:1 ratio, dominant to recessive, and in dihybrid crosses of a 9:3 to 3:1 ratio, with 9 being double dominant and 1 being double recessive. There are derivations from the expected that do not follow simple Mendelian patterns. The first of the variants is incomplete dominance. In simple form dominant, alleles are expressed in the presence of the recessive allele or not. Incomplete dominance, however, expresses an intermediate phenotype, kind of like the blending of alleles. This means that the dominant gene is not expressed if the recessive allele is present, and neither allele is dominant over the other. Incomplete dominance is seen often in flowers, where there are 3 variations of the trait color. For example, a white flower and a red flower are expressed in the homozygous true breeding forms, but when crossed, the heterozygous offspring are all pink. This heterozygous intermediate contains only partial expression of the dominant allele and partial expression of the recessive allele. When we sibmate the pink flowers, the phenotypic ratio becomes 1 red to 2 pink to 1 white, instead of the expected 3:1 following simple Mendelian patterns. In this case of incomplete dominance, the genetic phenotypic ratios and genetic genotypic ratios are identical. Let's look at an example of incomplete dominance. Let's say we have a pink flower and we want to cross it with a white flower. Would these parents be able to create red offspring? The pink genotype is big R little r, and the white genotype is little r. When we cross them, the Punnett square follows, with 50% of the offspring being big R little r for pink, and 50% of the offspring being little r for white. So these parents could not have red offspring. Conversely, codominance is exhibited when neither of the homozygous forms is dominant over the other. For example, a brown cow and a white cow mate, and have a cow with white and brown patches that are both expressed, or a pink and a white flower mate and form a flower containing a marbled combination of pink and white. Another form of codominance is seen in humans, and it is our ABO blood typing. Humans can express the blood type of either A, B, AB, or O. Neither A nor B are dominant over one another, but both A and B are dominant over O. So O is the recessive form. To understand blood typing, we look at the antigens on the red blood cells and see that antigen A is on type A blood and antigen B is on type B blood. No antigens are on type O blood, and type AB blood has both antigens, A and B. A person can exhibit type A blood with either an AA or AO genotype. The person with B blood can exhibit either a BB or BO genotype. A person with O blood must have an OO genotype, and a person with AB blood must have the AB genotype. This means that a person with type A blood can get either A or O blood donated to them, but they can give blood to either A or AB individuals. A person with type B blood can receive blood from type B or O individuals, but can only donate to people with B blood or AB blood. Individuals with AB blood can receive any type of blood, A, B, O, AB, all of it, but they can only donate to AB individuals. So AB is referred to as the universal acceptor. Type O blood is the universal donor and can be donated to any other blood type, A, B, O, or AB. But O individuals can only receive O blood. The antibodies found in A blood are anti-B and cause clotting. The antibodies in type B blood are anti-A, which can cause clotting. AB blood has neither A nor B antibodies, while type O blood contains both. One way to rule out paternity is through blood typing. Say a husband and a wife have a child with type A blood. Mom is type O and dad is type B. Could this child be dad's? No. Dad could either be the BB or BO genotype. He could only have type B children or possibly a type O child. Maybe he could contribute to an AB child, but he could never have an A child unless the mom was A. And he was BO. This mom is O, so she has to be OO, which means she can only give an O. She could not have an AB child or contribute anything other than O. So if they had an A child, her child would not be that father's because he had type B blood. This shows that paternity can be ruled out. He could not be the father based on the blood typing. However, blood typing is never used as a sole acceptance for paternity, as it is only a possibility that if a male father and a mother had a child, and the child had a blood type that could come from both those parents, it is a possibility that he could have been the father. Another derivation from simple Mendelian genetics is pleiotropy. In this case, one gene is affecting multiple traits. This gene can be expressed at various times in development or expressed for different reasons in alternate types of cells or body locations. For example, a mutation in the types of beta globulins can lead to blindness. They can lead to liver disease, heart attacks, and even sickle cell anemia. Sickle cell is a disease that is inherited through a recessive gene in which the hemoglobin is malformed. It creates blockages in the arteries and veins and hinders blood flow to the tissues and organs, which don't receive enough oxygenated blood. Why then, as a recessive disease, does this recessive allele still exist in the population? The answer is because of a selected advantage provided by the heterozygote form. The disease is commonly found in African Americans, and in Africa, malaria is highly prevalent. Heterozygote malformation of blood cells occurs only in half of their blood cells, or a smaller portion, which creates one portion of their blood that cells cannot grow and reproduce parasites in. This offers these individuals, with only a portion of their cells as sickle-shaped, an advantage of the survival from malaria. This survival advantage keeps the recessive disease within the population. Epistasis is another example of a non-Mendelian genetic inheritance pattern. Epistasis is where one gene affects another gene's expression. For example, in dogs, the coat color is controlled by a gene, where black is dominant over brown. In order for this gene to be expressed, another gene for pigmentation must be expressed. So if the pigmentation is expressed in dominance, then you can see the coat color. If it is not expressed, then you won't be able to see the black or brown coat color, and the coat will be blonde. In this case, E stands for epistasis, and B stands for black. So big E, or big E, little e, are both expressing pigment. Because of that, if the dog has a big B, it will be black. If it has two small b's, it will be brown. If, however, the epistatic allele is little e, it can only express a blonde coat. It cannot express the black or the brown. Temperature can affect gene expression as well. In Siamese cats, the expression of an albino recessive gene for melanin production only occurs at colder temperatures. As the cat ages, the protein is expressed to produce melanin. Extremities are colder than the body, which creates the darker pigments in the paws, ears, and face. Himalayan rabbits have a temperature-sensitive allele that produces pigments at lower temperatures. So if they're reared in a warmer environment, they'll be all white. Where if they're reared in cold climates, they will maintain the black on their paws, ears, face, and tail. If you shave the fur off a Himalayan rabbit and place an ice pack on its back, the fur would grow back and be black instead of white because of the colder temperature. Another example of the environment dictating when genes will be expressed includes plants. Even when genetically identical plants are grown, they will grow to different heights based on the temperature and environment that they're grown in. Of course, as with all living things, there's an average range that is optimal for plants to grow to the average height. However, the total possible growth of a plant is known as the norm of reaction. Geneticists cannot cross humans for study, partially because they have such long gestation periods and life cycles, on top of the ethical concerns, of course. Because of this, one approach to study human diseases we use is called a pedigree. With pedigree, variations and trends in the traits can be seen through multiple generations in a family through one image. The squares on a pedigree are male and the circles are female. Unaffected individuals have a homozygous genotype and are completely empty, where heterozygotes are half filled in and are usually carriers of the disease. Many carriers do not exhibit the disease traits or symptoms. Affected individuals have the opposite homozygous genotype and are completely filled in. Most diseases are autosomal recessive, such as cystic fibrosis and sickle cell anemia. So the colored individuals would be homozygous recessive. However, there are instances where the disease is autosomal dominant, in cases such as Huntington's disease. Let's look at a pedigree with family members in cystic fibrosis. Cystic fibrosis heterozygotes are carriers but do not express symptoms of the disease. Homozygous recessive individuals are affected with the disease. In this example, we would need to begin by identifying the genotypes of individuals that do not have the disease. Homozygote recessive is known for expressing the disease, so we can fill all those people in as little a. Then we know that all of those affected must have at least one big A allele. When writing a genotype with an unknown second allele, we use the underscore to hold its place. In the parental generation, we can see that one parent expresses the disease and the other does not. The parents have offspring with the disease, so the unaffected parent must be a heterozygous carrier. So we have an affected parent with little a, and an unaffected parent with big A that contribute to their children. If they're contributing to a recessive child, they'll have little a. And with a dominant child, then mom has the big A that she gave because the dad can only give the little a. In the offspring, in generation 2, there are 4 children attached to the T that connects the parental generation, or generation 1. These 4 children are labeled with a Roman numeral for generation. They would be generation 2-2, 2-3, 2-5, and 2-6. The other individuals in the second generation are their mates. This would include 2-1, 2-4, and 2-7. Looking at the first couple in generation 2, their child is affected, which means that parent 2-1 must be a heterozygous carrier in order to create an affected child with the affected father. The same is true for parent 2-4. They must be a carrier with one large A and one small a to pass on the disease to their daughter. When looking at the child 2-5, since the child is not presenting the disease, they must have gotten the dominant allele from mom, and therefore they must be a heterozygote carrier because dad could only give a small a. This would be true as well for the unaffected offspring in generation 3. Because the father exhibits the disease, the offspring must have one recessive allele from dad. Since they do not express the disease, they must have one dominant allele from mom. Another derivation from simple Mendelian predictions lies in mutations. When an allele contains a mutation, it may not be expressed. For example, a loss of function mutation could result in a recessive allele becoming expressed in the heterozygote form. It could also result in underexpression or hyperexpression of a protein. A gain of function allele mutation could result in genes that gain new functions. Conversely, it can also hypoexpress or overexpress the protein that it's coding for. Our last non-Mendelian genetic pattern is called X-linked, or sometimes referred to as sex-linked. In humans, each normal diploid individual carries a minimum of one X chromosome. This means that both males and females are affected by the traits carried on the X chromosome. Males are hemizygous, as they carry only one copy, while females can carry 2 copies of X, so they can be carriers as heterozygotes, or they can be homozygous dominant or homozygous recessive. In order for diseases to be manifested with symptoms, the female must possess 2 copies of the disease trait. Diseases occurring on the X-linked chromosome, such as hemophilia, occur in a disproportionate number of male offspring as compared to female offspring from the same parents. This is because males only need one copy of the X to express the disease. Hemophilia is caused by an X-linked recessive gene that severely reduces the body's capacity to stop blood flow through clotting. I like to use the X and Y alleles to identify male and female individual offspring. As well, I like to use capital H for dominant allele, lowercase h for the recessive allele in hemophilia. If the father is a hemophiliac, he would be X little hy. If the mother is a carrier, her genotype would be X big H, X little h. Each time this couple mates, they have a 25% chance of having a girl with hemophilia, a 25% chance of having a girl carrier, a 25% chance of having a male hemophiliac, and a 25% chance of having an unaffected male. We could look at it this way as well. If these parents have a child and it's female, there's a 50% chance of her being a carrier and a 50% chance of her being hemophiliac. The same idea with the sons. If this couple had a boy child, there's a 50% chance the son would be a hemophiliac and a 50% chance he does not carry the disease. He would not carry the disease on his alleles at all because he got the X from mom and his other allele is Y. It is very important that you read the questions very carefully to determine exactly what the question is asking for. Let's look at another example. If the mother is a carrier and the father is unaffected, we can see that they have a 75% chance of having an unaffected child and a 25% chance of having an affected child. If we look at the breakdown, we see 25% chance of having an unaffected female, a 25% chance of having an unaffected male, a 25% chance of having an unaffected carrier female, and a 25% chance of having a hemophiliac male. Again, the question might ask about a specific sex child, not just a child. If they have a female, there would be a 50% chance she's a carrier and a 50% chance she's unaffected. If they have a male, there's a 50% chance he will be unaffected and a 50% chance that he'd be hemophiliac. Regardless of whether the father is a hemophiliac or not, all sons will have a 50% chance of exhibiting hemophilia because mom carries it on the X chromosome. Students should be able to differentiate between codominance and incomplete dominance. Students should understand differences between epistasis and pleiotropy. Students should describe how you use pedigrees. Students should be able to discuss environmental influence on genes and gain or loss of function alleles. Students should also be able to determine the percent offspring with X linkage factors.